right, welcome to Lesson 2 of Introduction to Ecology. Today we're going to talk about evolution. And I realize many of you are taking an entire course on evolution. Some of you have already had it. Some of you will be taking it next semester. So obviously we're not going to cover everything about evolution in one lecture. Today my goal is to sort of teach you the basic ideas, basic foundational concepts, and in particular focus on those concepts that are most important for ecology and understanding how evolutionary processes relate to ecology. So last class we talked about the two sort of different perspectives on evolution, the two different ways we can define evolution. First, we said that evolution is the change in the genetic composition of a population over time. So this is a change in what we're going to call allele frequency. And so we talked about that common example where we had a population of primarily white moths with a few gray moths. And then the Industrial Revolution happened. We got a lot of soot on the trees. The white moths stood out. They got eaten by predators. The gray moths were much better camouflaged. They blended in better, and so over time, the gray moths survived and reproduced more, and eventually the population ended up with mostly gray moths and a few white moths scattered in. So that's one example of evolution. That's where we had a change in the genetic composition of the population occurring as a result of the selective pressures and changes in the selective pressures in the population. The other definition that we talked about was descent with modification, where we have accumulation of differences from an ancestral state. So here we have sort of the ancestral state where we had small moths with plain antennae, all white, kind of little, kind of boring. And then over time what we might see is that the antennae suddenly become very ornate. The moths get bigger. The forewings get enlarged. They turn gray and dotted. And so we have all these little changes, and eventually that ancestral state looks very different from the current state we have today. Even though those two organisms are related, even though those very ornate large moths are related to those tiny boring moths, so much change has happened through millions of years or hundreds of millions of years that now what we see are we've got sort of very different organisms, and that's also a process. It's also the process of evolution. So to understand evolution, we need to understand heritability and a heritable trait. And a heritable trait is any trait that's passed from parent to offspring. So a good example in humans would be natural hair color. Natural hair color is heritable. So if I have a mom with brown hair, chances are she's going to have offspring with brown hair. That's particularly true if dad also has brown hair. And so this is a heritable trait because moms with brown hair are more likely to have brown-haired children than moms with other colored hair. A non-heritable trait, however, is actual hair color. So if I have a mom whose natural hair color is brown, but who chooses to dye her hair so that she has blonde hair, blonde is not heritable, right? That's her actual hair color, not her natural hair color. And it's blonde only because she's gotten that out of a box. So it doesn't matter how much blonde hair dye that mom uses, she's still going to have brown-haired children, right? And so actual hair color is non-heritable because it's not something that mom can pass down to her kids, at least if she's used some kind of modification. So that mom can dye her hair pink, she can dye her hair green, she can dye her hair purple. When she does those things, she's not going to have pink-haired kids, green-haired kids, purple-haired kids. She's still going to have brown-haired kids because actual hair color is non-heritable. This is very important. Evolution can only occur on heritable traits. So if you do something to your hair or your nose, you have plastic surgery, you wear different colored lenses on your eyes, all that sort of stuff that you can do that changes how you look but can't be passed on, evolution can't work on that. It can only work on these traits that are passed on from the parent to the offspring. Now I've simplified a little bit. Hair color is very complicated. Not only does the dad's hair color matter, but in fact, hair color is controlled by a whole pile of different genes on different chromosomes. And so brunette parents can have blonde children, blonde parents can have brunette children, and part of that's because there's so many genes involved. And this is an example. This is my own hair color, and this is from 23andMe, where they've pinpointed all the different genes that are involved in determining your hair color. And the bigger the gene is, the more significant that particular gene is in determining hair color. and anything that's pink tends to be a brown gene that I've got, and anything that's yellow tends to be a blonde gene that I've got. And so you notice I have a lot more pink than yellow, and so it's kind of likely that I would be a brunette, and in fact I am. So yes, hair color is determined by your genes. It is a heritable trait, but it's very complicated because there's a lot of different genes involved, not just one. All right, so I've mentioned this word genes. Hopefully this is familiar to a lot of you. Genes are basically sequences of DNA, or deoxyribonucleic acid. And genes specify how to build proteins. So basically I have DNA sequences, and those specify what's known as amino acid sequences. And then the amino acid sequences are basically the blueprint for what the protein is. And so when I talk about genes, they're really just codes for proteins. And different genes make different proteins. And sometimes you have different gene variants at the same place in a chromosome, and those make different protein variants. And so that's what we call an allele. An allele is different forms of a gene. And so those different forms actually have slightly different DNA sequences. And because they have slightly different DNA sequences, they code for slightly different amino acid sequences. And depending on those differences in the amino acid sequences, those proteins fold up differently, they have slightly different shapes, they have slightly different functions, and as a result, that causes differences that we actually see across different organisms and different people. So a good example is the one of the genes that codes for the texture of your hair. And so we know that this gene has a couple of different forms. In one form, the amino acid sequence actually has a TAC, and in another form, at that same location, it has a TAG. So there's one base pair in that sequence that's different, and as a result, we actually have a different amino acid sequence in the TAC gene versus the TAG gene. And that different amino acid sequence results in slightly different shaped proteins. And those different shaped proteins actually cause changes in your hair shape. So the TAC codes for sort of a curlier hair, and the TAG codes for a straighter hair. Now, it's a little bit more complicated because most organisms, not all, but most, and definitely humans, have multiple copies of each gene, right? And so in humans, we have 2 copies of each gene. One comes from mom and one comes from dad. And so if I have 2 curly hair genes, 2 curly haired alleles, right, at a particular gene, then I'm going to probably have curly hair. If I have 2 straight hair alleles, I am probably going to have straight hair. But it's possible I got a curly hair from mom and a straight hair from dad, and now I've sort of got 2 different alleles, one at each of my 2 copies for that particular gene, right? And so that's where things get a little complicated. So 2 more definitions. We have genotype. Genotype is the genetic makeup of an individual at a particular gene. And what that means is how many of each allele does that person have, or does that organism have? And typically we represent this with letters. And so, you know, if you have 2 different alleles, we have a big A allele and a little a allele, then we would represent the genotypes as big A, little a, and little a, little a. If there's only 2 alleles, those are the only possible combinations you can have. You can have more than 2 alleles. You could have 3, 4, 5, 6 alleles. You're still only going to have 2 copies of each gene, but in the population at large, there may be multiple different alleles floating around. All right, so your genotype is how many of each particular allele you have. And with people, again, we're going to have 2 alleles, and they might be the same or they might be different. Phenotype is the observable characteristic of an individual. And so different genotypes may have different phenotypes. And one of the best examples is eye color in humans. And again, this is a bit of a simplification, but it's a good example. So if I have a person who has little b as my 2 alleles, that person has blue eyes. And blue eyes is the phenotype, right? And the genotype is little b. If I have a person that has a big B genotype, then they have a brown phenotype. However, people who have a big B genotype also have a brown phenotype. So there are two different genotypes, big B, and big B, little b, that give the same phenotype brown. And there's only one genotype, little b, that gives the phenotype blue. So again, phenotype is what it looks like. Genotype is the actual gene composition, the allele composition. So another example beyond human eye color is in squirrels and in squirrel coloration. So in gray squirrels, there's the MC1R gene that controls fur color. And there are at least two known alleles for this gene. And one of the DNA sequences is missing a chunk of amino acids. It's not even like we have one DNA base pair substituted. There's a whole chunk of amino acids that are just missing from one copy of that particular gene, from one form of that gene, from one allele. Whereas in the other allele, those amino acid sequences are present. And so you can see those dark gray amino acids in that folded up protein, those are the ones that are missing in the one allele and present in the other. So again, we have these two alleles. There may be more, but scientists know about two that are in the population. And as a result, because we have two alleles, we actually have three genotypes. We have big A, big A, little a, and little a, little a. And we call the two genotypes that have the same type of allele at each locus, we call those homozygous. So big A, that's two of the same, two big A's, that's homozygous. Little a, that's two of the same, so it's also homozygous. So those are homozygous genotypes. We call the genotype that has two different alleles, big A, heterozygous, because there's two different, that's what hetero means. All right. Now, unlike eye color in humans, where we had two different genotypes that gave the same phenotype, in this particular squirrel example, each genotype has a different phenotype. So the big A genotype gives the gray phenotype. The big A genotype gives a brown-black phenotype, and the little a genotype gives a jet-black phenotype. All right. So, anytime that allele frequency changes, evolution has occurred. That was one of our definitions. That was our first definition for evolution. So you might wonder, how do I know if allele frequency has changed? Well, the first thing you want to do is look at allele frequency at one time point, look at allele frequency at another time point, and say, are they different? If they're different, allele frequency has changed, and that means evolution has occurred. So I've got a particular population of squirrels shown on this slide, and there are 10 squirrels. And because at this particular fur color gene, we have two different alleles, and because squirrels have two copies of each gene, right? So we have a total of two different allele types floating around the population, and there are 20 allele spots to be filled up in this population. That makes sense. So each squirrel has two copies of an allele, and there are 10 squirrels, so there are 20 alleles. All right. So now what I want to do is I want to calculate the allele frequency at one time point. Let's take a look at the number of big A alleles. Each of my gray squirrels has two big A alleles, and there are 5 gray squirrels, so that gives me 10 big A alleles. Each of my brown squirrels has 1 big A allele, and there are 3 brown squirrels, so that gives me another 3 big A alleles, for a total of 13 big A alleles. So in this population, I have 13 big A alleles. But that's not the frequency, that's the number. If I want the frequency, I have to divide by the total number of alleles. And again, we said 10 squirrels, each has 2 alleles, so that's 20 alleles. So I divide 13 by 20, and I end up with 0.65. So that's the frequency of big A alleles. I can do the same for little a alleles. I have 2 black squirrels, and each of those comes with 2 little a alleles, so that's 4. And I have 3 brown squirrels, and each of those comes with 1 little a allele. So that's another 3. So that is 7 little a alleles. Again, so that's the number of little a alleles in this population. If I want the frequency, I have to divide by the total number of alleles, which is 20, and that gives me 0.35. So that's my frequency of little a alleles. And so basically I have 65% big A alleles and 35% little a alleles. Now, if I come back 20 years later and I notice that I've got a lot more black and brown squirrels, That's going to give me a hint. Maybe the allele frequency has changed. But to know for sure, let's figure out what the new allele frequencies are. Let's figure out if they've changed. All right, so if I actually add this all up, and you can do this at home, what I find out is that I have 40% big A allele, and I have 60% little a allele. So if you remember, I had previously 65% big A, and now I've gone down to 40%. And I previously had 35% little a, and I've gone up to 60% little a. And again, practice yourself, see that you get those same numbers. But assuming I've calculated everything correctly, I've observed a change. I've observed a change in the big A alleles and the little A alleles. I've lost big A alleles. I've gained little A alleles. That means that evolution has occurred. All right. If a population is not evolving, in other words, if it has the same allele frequency from one generation to the next, then it's said to be in Hardy-Weinberg equilibrium. Equilibrium means something is not changing. It's constant all the time. And so when a population, and only when a population is in Hardy-Weinberg equilibrium, you can actually predict genotype frequency based on allele frequency. Remember, allele frequency is the frequency of each particular form of the gene. Genotype frequency is the frequency of particular genotypes, which are, the makeup, the genetic makeup, the number of each allele that each individual has. All right. So to do this, a few definitions. We're going to say little p is the frequency of one particular allele, in this case the big A allele, and we're going to say little q is the frequency of the second allele, or the little a allele in this particular example. Once we define those, for a population that's in Hardy-Weinberg equilibrium, we know that the frequency of the big A genotype is p squared. We know that the frequency of the little a genotype is q squared. And we know that the frequency of the big A genotype is 2 times pq. All right? Again, that's only true if the system is in Hardy-Weinberg equilibrium. If the system is evolving and changing over time, that is not true. All right. Now, sometimes we can actually go from the frequency of the genotype to the frequency of the phenotype. That's really easy in the case where each genotype has a distinct phenotype. So with the squirrels, each genotype gives you a different phenotype. And so we know that p squared is not only the frequency of the big A genotype, it's also the frequency of gray squirrels. Q squared is not only the frequency of the little a genotype, it's also the frequency of the black phenotype. And 2pq is not only the frequency of the big A genotype, it's also the frequency of the brown phenotype. That's not always true when you have multiple genotypes giving you a particular phenotype, okay? So it just depends on the scenario what exactly you can figure out. Sometimes you can, sometimes you can't. Sometimes you have to think about it, and we'll give some questions like that. Okay. So one way to ask questions about Hardy-Weinberg equilibrium or populations that are in Hardy-Weinberg equilibrium is to ask, is this particular population in Hardy-Weinberg equilibrium, right? How do you know? Well, you basically need to figure out if the frequency of genotypes you're seeing matches the frequency you would expect based on the frequency of alleles. All right, so in this particular population, we have 40% big A alleles and 60% little a alleles. Again, I didn't work that out. I told you to do it, so if you don't believe me, try it. Tell me I'm wrong. Make sure you know how to do that. So now we know what the frequency of the alleles are. Let's figure out if we're getting the right frequency or the right number of the different genotypes. So let's take a look at the big A genotype, which is also the gray phenotype. We said we had 40% big A alleles, so that means P is 0.4. And if we have a 0.4 frequency for big A alleles, that means P squared should be the frequency of the big A genotype. In other words, we take 0.4 times 0.4, and that's 0.16. So that gives me the frequency that I would expect for the big A genotype, which in this particular squirrel example is also the gray phenotype. Since I had 10 squirrels in my entire population, the number of gray squirrels I would expect based on this particular frequency of gray squirrels is 10 times 0.6. That gives me 1.6 squirrels. I would never expect a part of a squirrel, right? So if I expect 1.6 gray squirrels, that means somewhere between 1 and 2 gray squirrels is what I expect in a population of 10 gray squirrels, or a population of 10 squirrels total. And that's what I see. I've got 2 gray squirrels. All right, now let's take a look at black squirrels. So again, the frequency of the little a allele is 0.6, and that means the frequency of the little a genotype, which also happens to be the black phenotype, should be q squared, so 0.6 times 0.6, that equals 0.36. Again, since I have 10 squirrels in the population, I want to take 10 times 0.36, and that gives me 3.6. As before, I don't expect a part of a squirrel, so if I would on average expect 3.6 squirrels to be black in a population of 10, that means anywhere from 3 to 4, and I have 4, so that matches. Yep, all good so far. Now let's take a look at the heterozygous genotype. So again, we have 0.4 is our frequency of the big A allele, 0.6 is our frequency of the little a allele, and remember we said that the frequency of the heterozygote in a Hardy-Weinberg equilibrium population is going to be 2 times 0.4 times 0.6, or 0.48. Again, I had 10 squirrels in the population, so I'm going to take 10 times 0.48. And what that means, again, I would on average expect 4.8 squirrels to be brown, but again, we don't have parts of squirrels, so anywhere from 4 to 5 squirrels should be brown, and that's what I see. So my answer would be yes, this population does look like it's in Hardy-Weinberg equilibrium because the frequencies of my genotypes match what I would expect based on the frequencies of my alleles. So you can ask Hardy-Weinberg equilibrium questions in other ways. Right now, we're going to do an example in class. For those of you who did not come to class and are just watching this online, I would suggest that you try and work out this example for yourself. And if you can't do that, I would strongly suggest you find a more conscientious student in the class and borrow their notes. Make sure you know how to do this. So again, you can expect Hardy-Weinberg equilibrium questions to be on the exam. Make sure you know how to do them, especially if you didn't see this worked out in class. All right. So we've talked a lot about shifts in allele frequency across populations, and when we look at that, we can talk about Hardy-Weinberg equilibrium in populations that have stopped shifting and have sort of reached that equilibrium point. And that was our one definition for evolution. Our other definition for evolution was descent with modification. And this was a term that was first used by Charles Darwin in The Origin of Species. And he used it for a couple of reasons. So he used the word descent because he wanted to get across that idea that these sort of ancestral states and these modern versions of these organisms, they have a shared ancestry. They came from, the new current modern species came from these ancestors. And as a result of that, they share characteristics with the ancestors. However, at the same time, because of selective pressures, because of sort of these differences that are accumulating over time, over many, many years, they're also different. And so they're modified. So it's that idea of descent where they have some characteristics that are shared with their ancestors, and modification where they have some characteristics that are different, that have built up differences over time. And that's that idea of evolution. They come from the same past, but they're also different from the past. So Charles Darwin hypothesized that descent with modification occurred because of a process known as natural selection. And the idea behind natural selection is that individuals with certain heritable characteristics, remember evolution only works on heritable characteristics, individuals with certain favorable heritable characteristics survive and reproduce better than their other organisms in the population. And as a result, they leave more offspring to the next generation, they do better, and over time, that particular heritable characteristic gets overrepresented in the population, while the heritable characteristics that were not so good, that were not so favorable, that were not selected for, kind of die out. And so the idea is that over time, as different selective pressures sort of favor this trait over that, individual animals build up these differences, individual populations, and then they become very different from their parents or their ancestors. And eventually they become so different that they're no longer anything like their ancestors. They're actually a new species. All right, so how do populations diverge? Generally, in order for populations to diverge, and that means become different from one another, they have to experience different conditions. There has to be selection favoring one set of heritable traits for the one population, and a different set of heritable traits for another population. And this happens very frequently. I like this particular example because it's sort of happened over a very short time scale. But many of you have probably read the story of the very quiet cricket. It's by Eric Carle. And this is about a cricket who just doesn't know how to sing or can't make a sound to attract mates. And that may seem like fiction, but in fact, there is a place where some of the crickets can't sing. They're very quiet. And it's actually on the Hawaiian Islands. So there is a type of field cricket that's present on all the Hawaiian Islands, and it's been there for many, many, many years. And historically, all of these field crickets used to sing, and they used to sing in order to attract mates. That was the way that the males made some sort of sound that the females could cue in on and find the males. Now, in the 1900s, a parasitic fly got introduced to the Hawaiian Islands. And this parasitic fly actually lays its eggs in the crickets, and the fly larvae basically eat the cricket from inside and kill it. Now, that's great, but what these flies use to find the crickets is actually their song. And so the crickets that are singing to attract a mate actually end up attracting these parasitic flies, and the parasitic flies kill them. It's a little sad. So these flies were first introduced on Kauai, spread down to Oahu, and then to the Big Island. And they've actually built up to very, very high densities on Kauai. And so researchers went in the early 2000s, I believe, and what they found is that many of the crickets on Kauai weren't singing anymore. And in fact, they found that the crickets on Oahu also weren't singing. And when they got to some of the other islands that were not parasitized by the fly, or that the fly wasn't present, the crickets still sang. And so what they were observing was different selective pressures on the different islands. On the islands that had the parasitic fly, there was a strong selective pressure for the crickets not to sing. Because even though they needed to sing to attract a mate, to find a mate, the singing was deadly. And so evolution selected for crickets that didn't have the machinery, that didn't have the wing structure to sing. That wasn't the case on the islands that didn't have flies. Now, what they found really high densities of very quiet crickets on Kauai and pretty high densities on Oahu. The parasitic fly has also been introduced to the Big Island, but it's at much lower densities. It doesn't seem to do as well on that island. We don't know why. And they found that some of the crickets on the Big Island don't sing, but there's actually a number that still continue to sing because the density of the flies is much lower. So again, we have no singing on Kauai, very, very little singing on Oahu, still quite a bit of singing on the Big Island because the parasitic flies are so low in density, and we have full singing on all the rest of the islands where there are no parasitic flies. So this is an example where the crickets on the islands with the parasitic flies actually evolved wing shapes that just can't sing. They can't make any sound. And so basically the crickets that couldn't sing as much, even though they couldn't attract a mate, did better. They produced more offspring because they weren't killed immediately by these parasitic flies. So you might think that that only happens with crickets or insects or, the wild, out in the wild, but it actually happens with humans too. And so if we think about humans, Europeans have been domesticating cows and using cows for milk and cheese and that sort of thing for many, many years. But other cultures, that's not so common. That's not as common in many cultures in Africa, and it's also not common in a lot of Asian cultures. And so there's a benefit, or there was historically a benefit to Europeans if their kids were able to drink milk, because milk was a pretty steady source of food. You had the cows ate grass, which was not a great source of food. The cows made milk, and that was a great source of food for you. So if your kids couldn't drink milk, they may not survive the winter. But if they could drink milk, they were probably good. So in European countries, the ability to drink cow's milk actually led to improved survival and improved reproduction. That was not true in other populations in other places. So in China, keeping cows was not very common, and so the ability to drink milk didn't really matter. Sure, you could drink milk, but if you were never given milk, who cares? And so in those countries where cows were not farmed, there was no real benefit to the ability to drink milk. And so what we find is that 77% of European Americans have alleles at two different genes that are associated with lactase persistence, which gives you the ability to digest and use milk as a food source. However, in China, across all of China, almost no people have these alleles. And again, that's because of those different selective pressures where European populations were cultivating cows, whereas Chinese populations were living on different food sources that required sort of different enzymes and different abilities. So there's 4 different mechanisms that are important for making evolution happen. There's 4 different processes. The first is mutation, and this is basically where you get a change in DNA sequences. It's just sort of a random change. It might be a substitution from one base to another base. It might be a deletion where you're just removing a whole chunk of bases. You can have a pile of different types of mutation, but in the end, they're just sort of random changes to your DNA. Then you can have natural selection, and we talked about this. This was Darwin's idea, where organisms that have beneficial heritable traits, like being gray, if you're, trying to blend into a gray background, you do better, and so you survive better, and you reproduce better, and you leave more offspring to the next generation. There's also genetic drift. And this is a stochastic process where, you can have great genes. You can just be like absolutely the best for your environment. You can have great chances of reproduction and survival because you do everything right and you have the right heritable traits to make you really successful, and a moose comes along and steps on you. And it doesn't matter how good your genes were. If the moose stepped on you, you're still dead. So that's the idea of genetic drift. And the 4th process that's important for evolution and how evolution happens is gene flow. And that's where we have sort of organisms or gametes from one population with sort of certain alleles drifting into another population. So here we might have gray squirrels on one side of a mountain, black squirrels on another side of the mountain. We have all big A alleles on one side of the mountain. We have all little a alleles on the other side of the mountain. And all it takes is for that one black squirrel to climb the mountain and go over the other side, and then we have mixing between the populations. And so that's gene flow, where we have sort of new alleles introduced because of movement. So we're going to go into each of these in a little more depth. Like I said, mutation is the change in your DNA sequence, the change in sort of the DNA that makes up a particular gene. And it happens because of copying errors and because of mechanical damage. And so there's a baseline rate that this happens no matter what, right? So that baseline rate is different in different organisms, but in humans it's pretty low. And that's not surprising because a lot of mutations are just bad. They might be deadly. They, silence some gene that you needed to live. They make some gene that makes something that's toxic or deadly, right? A lot of them are bad, but sometimes they're good. And so typically across populations, there's a baseline rate. This just happens, and it leads to mutations. Sometimes we up the rate at which mutations happen, and one of the best ways to do that is by exposure to high energy radiation. Whether you're, out exposed to UV from the sun or X-rays or some kind of nuclear blast, all of that actually causes damage to your DNA, and when the DNA gets repaired, sometimes it doesn't get repaired right, and that leads to a mutation. Mutation is really the only way to make different alleles. When we have different alleles in a population, those alleles arose as a result of mutation, because each allele is a different form of the gene, and what that means is the amino acid sequences are different, and what that means is the DNA sequences coding them are different. And so the only way you can get multiple different alleles is to have mutation give rise to new alleles. And so mutation provides the raw material on which all evolution is based. However, because many mutations are bad, because they frequently lead to sort of lethal effects, they tend to occur at a very low rate. There's sort of been selective pressures to make sure mutation isn't happening too fast in most organisms. And so in humans, we tend to find that mutations happen about 10 to the minus 4th to 10 to the minus 6 new mutations per gene per generation. So if we assume that humans have 20,000 to 25,000 genes, that means that one or two mutations, you may have 1 or 2 mutations that you did not inherit from your parent. In other words, there might be 1 or 2 genes where there's something slightly different that didn't come from dad and didn't come from mom. It came from a random miscopy when mom's egg got made or when dad's sperm got made. Right, so very, very few. So then you might think, well, I don't look anything like my mom. I don't look anything like my dad. Probably that's a good thing. And so you might think, well, why am I so different from mom and dad if I only have 1 or 2 mutations that are different from them? Well, there's 2 reasons. The first is that you only get 1 copy of each chromosome from your parent. So mom has 2 copies of each chromosome, but she only passes 1 on to you. Dad has 2 copies of each chromosome, and he only passes 1 of them on to you. And so right off the bat, you're missing half of mom's DNA, and you're missing half of dad's DNA. So right off the bat, you're going to be different. But the story's a little bit more complicated because of a process called recombination. And so mom had 2 copies of each chromosome, but she didn't just randomly give you 1 copy wholesale. What happened is her chromosomes actually kind of mixed and matched through this process of recombination where you reshuffle. And so you take, 1 piece from 1 chromosome and put it on the other chromosome before mom decides to give you one half of her, chromosomes. And so those 2 processes, recombination and as well as the fact you only inherit 1 chromosome from each parent, that's what makes you different from your parents. And so just to put this in perspective, the top picture is a picture of me when I was about 1 year old, and the bottom picture is a picture of my daughter when she was about 1 year old. And what you can see is that we're very similar. And part of that's because there aren't a lot of new mutations. She didn't suddenly develop, green ears because there's not that many mutations. But we're also different, and we're different because she only got half of my chromosomes and the other half came from dad. So I look at those and I say, oh, I see how we're different here or there, and I recognize that as coming from her dad or my husband. All right, so that's mutation. Moving on to the second process, natural selection, there's sort of 3 different ways the natural selection can go. Again, natural selection is that favoring of certain heritable traits so that organisms that possess those traits do better, produce more offspring, survive longer, and contribute more to the next generation. So, I can think about natural selection as selecting in one direction. So if I select for tails, for instance, maybe short tails are terrible, medium length tails are kind of okay, and long tails are really great, right? So basically the longer the tail, the more you move in that direction, the more you move towards long tails, the better. That's what's known as directional selection, because good is at one end of the spectrum and bad is at the other end of the spectrum. And it just keeps getting better as you move towards the one good direction, right? So with directional selection, you are selecting for an extreme trait, and you're selecting against the other extreme trait at the other end of the spectrum. Another possibility is what's known as stabilizing selection. So in this case, short tails are bad, but long tails are also bad. And what's best is right in the middle, right? Kind of like Goldilocks and the 3 bears. What's right is, what was sort of most favorable is that middle value. And so you don't want a really short tail, you don't want a really long tail, you want a medium length tail. You select against both of the extremes, both ends of the spectrum, and you select for the middle of the spectrum. That's again called stabilizing selection. Finally, we can have disruptive selection. And in this case, you're actually selecting for extremes, but it doesn't matter which. Short tails are great, long tails are great, but what's terrible is the medium length tails. Okay, so you're selecting against the middle, you're selecting for either one of the 2 extremes. And that's called disruptive selection because you're sort of disruptive. You want to go one way, you want to go the other way, you just don't want to be in the middle. And so you kind of get pushed in both directions. Now, if you have really strong selection for a particular allele, what you can find is that you end up with a population that's almost 100% of that one particular allele. And when that happens, that only happens because that allele is so favorable, so strongly favorable relative to everything else, that it basically sweeps through the population, and the organisms that have that allele just do amazingly, and they just completely displace all the other alleles and drive them to extinction. And when that happens, that's called fixation. When an allele reaches 100%, so that every single organism has 2 copies of that allele, that allele frequency is now 100% because there's no other allele in the population at all, and we say that allele has reached fixation. So this isn't super common, but a good example is in drug resistance in the malaria parasite. So what happened, we had malaria parasites, and before we started using our anti-malarial drugs, having resistance genes wasn't particularly beneficial. But when we started bombarding these malaria with the malarial drugs, all of a sudden having a resistance gene was really, really beneficial. Having that allele that conferred resistance to the drug we were bombarding really gave you an advantage in terms of leaving offspring. And so in 1997, when we had just started using the antimalarial drugs, we saw that only about 20% of alleles had this resistant phenotype. However, by 2005, what you see is this sweep where almost every single allele in the malaria parasite for that particular resistance gene is resistant, right? And so what we say is the drug resistance in malaria has gone to fixation. In other words, there are no malaria, you can't find malaria parasites out there that have even one copy of that allele that's non-resistant, that you can kill with the drug. So this happened with malaria. It also is happening with a lot of bacterial populations where we're using antibacterials because we put such strong selective pressure on these organisms by hitting them with the antibacterials. And so there's a huge fitness benefit to being resistant. And so those resistance alleles just sweep through the population and go to fixation. So that's natural selection. That's the second evolutionary process. The next evolutionary process is genetic drift. And like I said, this is stochastic or random or chance events. And because it depends on these random events, it's really mostly important in small populations. So if I have, 10 flowers and 5 of them get stomped on by a moose, that can dramatically change my population, right? Especially if the moose happens to stomp on the 5 good ones. But if I have millions and millions and millions of flowers, then what's the chance that the moose is going to stomp on all the good ones? Very unlikely, right? And so that's why genetic drift is usually only important in very small populations. That's a bad thing because typically organisms that are on their way out, that are being driven to extinction, exist as small populations. And so genetic drift becomes very important to these populations. So now I'm going to play a game that shows you how or demonstrates that importance of genetic drift and random processes in small populations. I'm not actually going to go through that online. So hopefully you were in class, hopefully you saw that. If not, just realize that what I'm showing right now is a game that we played to demonstrate the process of genetic drift and to show how, in large populations it's not such a big deal. In small populations, it's a really big deal. All right, so because of genetic drift, because of these sort of chance events in small populations, 4 things can happen. The first is that we can get massive fluctuations in allele frequency. If a moose stomps on 5 out of 10 of your flowers and you only had 10, then we can sort of go from one allele being 50% of the population to one allele being 100% of the population just because the moose stepped on the other half of the population. That can be a problem, and that can also lead to extinction and fixation. The allele that the moose happened to step on can disappear because it might have been a great allele, it might have been really helped with survival, but it just didn't protect against the moose because nothing would. And so that allele can go to extinction, while the poorer allele actually goes to fixation because it's the only one left after the moose has stomped on it. And so we get these massive swings in allele frequency in these small populations. Because of that, because we tend to have extinction being more likely in these small populations with genetic drift, we can actually get reduction in genetic variation. And that's because extinction is forever. Once it's gone, it's gone. You can't get it back. And so if you keep losing stuff, then you're just going to end up with less than what you started with. And so if you keep losing genetic variation because alleles just by chance happen to go extinct because a moose stepped on them, then you end up reducing genetic variation. And again, if you think about a population that's on its way to extinction anyways, that's a bad thing because genetic variation typically leads to robustness of a population. So the minute you start into this sort of vortex of losing genetic variation and getting smaller populations, you're kind of on your way out. Another bad thing can happen. You can actually get an increase in the frequency of harmful alleles. So usually, if natural selection is working properly on a large population, more beneficial alleles, good heritable traits, will go up in frequency because those parents survive better and leave more offspring. And bad heritable traits, harmful alleles, go down in frequency because the organisms carrying those traits survive more poorly, leave fewer offspring. But unfortunately, when we're in a small population with genetic drift, again, if I step on a couple of the good alleles, I can end up with the harmful alleles going up in frequency just because I didn't happen to step on those. And again, what that causes is an increase in these negative harmful alleles that in a larger population would have just been weeded out by the sort of likelihood of the better, more profitable alleles surviving better and producing better. We don't have that in small populations. And so even in humans, frequently in Ashkenazi Jewish populations, they went through a bottleneck where there was a very small population for a while. And as a result, some of these harmful alleles went up in frequency when they wouldn't have in a larger population. And so when you do genetic testing, frequently these are offered specifically to Ashkenazi Jewish populations because they went through that period with a very small population that allowed some of these harmful alleles to take hold and go up in frequency when they wouldn't have otherwise. Finally, genetic drift can actually lead to increased differences between populations. And again, that's because chance events in small populations can cause one allele to go up in frequency on one place and one allele to go down in frequency in another place, even if the environments in those two places are similar, even if there's no difference in terms of the selective pressures, but just chance happenings on one island were different from chance happenings on another island, and so those two different islands ended up diverging because of those sort of stochastic random events. And a good example is Channel Island foxes. So the Channel Islands are a set of islands that are just off the coast of California, but they're far enough from the mainland and they're far enough from each other that you don't have a lot of foxes swimming between the islands or between the island and the mainland. And so they've actually done studies, and you have different species of fox on each of the different islands. Now, these islands are not sort of far enough apart to be environmentally very different. There's no difference in predators or prey on the different islands. And so the hypothesis is that these different foxes are different primarily because of genetic drift, primarily because, the small population that colonized one island happened to be slightly different or happened to have slightly different early events happen when they were colonizing that island than the population on another island. And so over time, those differences built up just because they're small populations subject to these random fluctuations and random happenings. And as a result, we ended up with different species. So the final process that affects evolution is gene flow. And like I said, with these Channel Island foxes, there's not a lot of foxes moving between islands. If there were, we would have a lot of mixing, and so you wouldn't be able to get one species on one island and one species on another island because they would just be mixing and interbreeding the whole time. And so gene flow kind of acts against divergence of populations. It acts against speciation. Gene flow is basically the transfer of alleles from one population to another, whether that's across an ocean from one island to another, or whether that's across a field from one field to another, or whether it's across a mountain range from one side of the mountain to the other. So this movement can be with individuals, like a gray squirrel that brings two little A alleles over the mountain, but it could also be with gametes, something like pollen. And if you know anything about plants, the pollen is not sort of a full cell. It's more like the sperm in humans. It's only sort of the one half, but that's the dispersal. That's what moves. And so you can actually get gene flow from gametes blowing across. If I think about pine trees and the pollen that coats everything in the spring here, right? That's sort of the male sort of gamete floating through the air and bringing alleles with it as it goes. So gene flow is basically homogenizing. It mixes everything together. It makes populations more similar. And so the more gene flow you have across two areas, the more similar those populations are going to be. The less gene flow, for instance, if an ocean prevents foxes from moving, the less gene flow, the more dissimilar those populations are going to be. And again, the purpose of gene flow, or the effect of gene flow, is to introduce new alleles into a population. So this can be good. It can also be bad. If I think about genetically modified organisms, say I'm a farmer and I'm planting a field of genetically modified corn on one side, and a different farmer, and I'm planting an organic non-GMO field on the other side, it can be very hard to stop the pollen from the GMO field from drifting over and bringing those GMO, those genetically modified alleles, into my field. So again, gene flow can be helpful for populations because it can sort of prevent against some of these issues with small populations and genetic drift, but it can also be negative if those genes that are coming in are something you don't want, like genetically modified alleles. So the final idea I want to talk about is adaptive evolution. Adaptive evolution is basically a change in which traits that confer survival or reproductive advantage tend to increase in frequency over time, right? So in other words, things that are better for you, things that help you survive, do better in that environment, those should be going up, right? Because organisms with those traits tend to have more babies, they last longer, they contribute more to the next generation. And traits that are negative or at least not beneficial, that the organisms holding those don't survive as long, don't reproduce as much, those should decrease in frequency over time. Now, if we think about the four processes that we've talked about contributing to evolution, if I think about mutation, that's random, right? That's not going to cause an increase in frequency over time of any particular beneficial trait, right? Because it's just, there happened to be a copying error, or there happened to be a loss of a chunk of your DNA. It's a random process. It doesn't work to improve the ability of a particular organism to survive. Sometimes it goes the other way and it hurts the organism's survival. Natural selection, however, is not random, right? With natural selection, organisms that have beneficial traits live longer, have more babies, contribute more to the next generation. Organisms that do not have beneficial traits, that have negative traits, die, leave less babies to the next generation. And so natural selection is not random. Natural selection actually is a process that causes advantageous alleles to increase in frequency and disadvantageous alleles to decrease in frequency. Genetic drift, we talked about this. This is a completely random process. This is me stepping on some flowers and not other flowers. It had nothing to do with the fact that those flowers had beneficial traits or deleterious traits. It just had to do with chance and where I happened to put my foot. So again, that's a random process. Gene flow is also random. There's not necessarily any difference in the pollen that gets blown here versus blown there. It's just where the wind's blowing and what happens on that day. And so gene flow, again, doesn't tend to cause an increase in frequency of good beneficial traits and a decrease of bad negative traits. Gene flow just mixes everything up and says whatever happens, happens. So again, it's a very random process. So what I want you to notice is natural selection is the only evolutionary mechanism that consistently causes adaptive evolution, that consistently causes advantageous traits to increase in frequency and disadvantageous traits or bad traits to decrease in frequency. All right, so that's our summary of evolution. And if you have any questions, shoot me an email. And remember, don't put the G in there. It's just sbewick@clemson.edu. And again, hopefully you enjoyed our introduction to evolution. And again, it was a very brief introduction, but hopefully provided the basics for what you'll need for the rest of this course in ecology.